start sharing my slides. Can you see the slides well, Victoria? Fantastic. So thank you all very much for joining this morning to hear about my and my research group's focus on using AI to improve our understanding of resistant bugs. So with this, I obviously mean any sort of microbes that are resistant to especially antibiotics. And the title of my talk today is Squiggles to the Rescue, which I will explain right away on the first slide. So as Victoria already said, I'm affiliated with the Helmholtz AI Institute in Munich, so the core unit of the Helmholtz AI Institute, and also with the entire Computational Health Center there, as well as with the Technical University of Munich. And I started my research group there in 2022. The research group still sits in Munich, so if you want to talk to any of them after the talk, and you are based in Munich yourself, I will add names, and I added names and pictures. But yeah, this year I started a professorship at the University of Zurich. But we are still very keen on collaborating with all the AI researchers in Germany, because the focus of the professorship is more on the application of genomics in the context of One Health, which I will also explain in one moment. So yeah, let's get started. So when I talk about squiggles, I'm referring to nanopore sequencing technology and how we can use AI to understand the respective genomic data. So nanopore sequencing of DNA or RNA works by guiding single-stranded nucleotides through nanopores embedded in a membrane, which you see visualized here. And these nucleotides will then disturb an ionic current that is also going through these nanopores in a very characteristic manner. And this disruption of this ionic current is the so-called squiggle signal. So it's a really dirty signal, and used to be difficult to interpret until the advances in AI have made possible to like really accurately retrieve information from the squiggle data. The most important information that we want to retrieve is the nucleotide sequence, either of DNA or direct RNA sequencing. And we can now just apply like really tailored, improved base calling algorithms that are based on AI to this data and retrieve like with really high accuracy our nucleotide sequence. Now, this mode of sequencing is really unique in its way of working, in how it works, because we are actually looking at the native DNA and RNA, which has a lot of advantages. So firstly, we can sequence any sort of nucleotide fragment length. So we can obtain very short sequencing reads as well as really long sequencing reads. And we are talking multiple megabases here for those of you who know how significant this is. Basically sequencing reads that are as long as our original nucleotide fragments. And this is important because if we have like very long sequencing reads, we can use this to piece them together into so-called de novo genomes, and also, for example, de novo plasmids. So we can just use our raw data to obtain information about which microbes are present, and in understanding the pathogens and antimicrobial resistances, and if the resistances are, for example, encoded by plasmids, which I will talk about a little bit later. Because of how nanopore sequencing works, we can also do real-time data analysis. So basically at the very moment that the ionic current is being disrupted, we can base call this squiggle data into the respective sequence. And if we pair this with the option of doing portable sequencing, here you see an example of the portable sequencing device using the nanopore technology, the Minion device you might have heard of, paired together with some NVIDIA embedded AI machines that are also portable. We can really think of bringing genomic technology at the point where it's of interest, for example, at the point of care in the clinical setting, especially if we pair this with the low upfront investment costs, which means we can consider really having a genomic technology as accessible technology globally, also in low and middle income countries. And finally, nanopore sequencing is really exciting since we can leverage what I started calling the power of the squiggle to obtain a lot more than just information about the nucleotide sequence, which we would also be able to retrieve from other sequencing technologies. So I think what has become probably the most applied and most famous approach is to call epigenetic modifications, such as methylation that you can see here of the nucleotides from this raw squiggle data, because obviously these methylations are also going to change the squiggle data in a very characteristic manner. But there are also a lot of other things, I believe, other information that we can retrieve from this squiggle data, and I will talk a little bit about this later. Importantly, as I mentioned at the beginning, this sort of squiggle data is obviously very noisy. And like such factors such as methylation, for example, are introducing additional noise into the data. Plus, for a long time, the nanopore sequencing technology had problems with like a certain sequence content. For example, homopolymers, where a lot of bases would be, the same base would be repeated several times. The sequencing approach had problems because basically they couldn't distinguish like how many bases were in the pore at the same time, because in the end you can imagine this as a sliding window approach. But this has improved a lot, I would say, just in the last 1 or 2 years. And this was partially because of improvements in the technology. One of them, a really easy one, to make the pore longer, so more sensitive to what is inside. But also, obviously, due to the improvements in AI models. I think especially the application of transformer models to the squiggle data has really allowed us to obtain like really super high accuracy from this data. So I just want to emphasize that the technology together with AI now has allowed us to really make this applicable in a highly accurate manner. But let me show you some of these applications of nanopore technology and AI now. So in my group, which I said still sits in Munich at the moment, but which will also grow now in Zurich, we are in general interested in rapid One Health surveillance. With One Health, I mean this concept that health is more than just the health of humans or global health, but it extends to the health of all organisms and also the health of entire ecosystems. So it's a very complex system. Concept, but I think it makes a lot of sense if we think of examples such as zoonotic diseases or antimicrobial resistance, where we know that there's a clear link between human health and also the impact of humans onto the environment and the occurrence, for example, of pathogens or resistance genes in animals, can be wildlife or livestock, but also in the environment, such as in the air or in fresh water. So if we want to actually think about using nanopore technology and AI for surveillance, we obviously have to think about, we have to compare the surveillance approach to what we are currently in an established diagnostic manner using for surveillance. And this surveillance, and I'm taking like the food safety and the human health space, so the clinical application as an example here, are mostly based on culturing bacteria. So you obtain a sample that can be a patient sample, any sort of swab. It can be an environmental swab. It could be just water, air, or whatever. And you apply different culturing approaches to culture different sorts of microbes. So you incubate the sample, then you subculture all sorts of different potential pathogens that you might get. And you are subjecting those final cultures to what is called multitoph, which is a mass spectrometry approach that is being used to obtain the species of the microbe. And then you do resistance testing by the so-called YTEC2 approach, which is basically just exposure of these different cultures to different antibiotics at different concentrations to understand like how resistant they are. You often do some additional testing, genetics testing, to actually see where these resistances come from. So to actually really identify the genes that are responsible for that. So as you can see here, this approach can take very long. We just showed like sort of the average time it can take. For some microbes, it can take even longer, like several weeks and so on. And it's obviously quite laborsome. You need access to certain biosafety laboratories. And in addition, I think what's important is that we know that we can only culture like a probably small part of the microbiome out there. So a lot of microbes and potential pathogens or potential commensal carriers of resistances might be out there that we cannot actually culture by our current methods. Like here, we now think it is very promising to apply the nanopore sequencing technology approach. And what we first did in our research was to say, okay, we have to directly be able to compare nanopore technology with the established diagnostics. So what we do first is we take the genomes of these cultured microbes here at the very end of the subculturing, and instead of applying this really lengthy, what we call phenotypic testing, we apply our nanopore sequencing. We extract the DNA, we prepare the library, and then we sequence and in real time can obtain first sequencing results and interpret them. So I am going to show you some examples of research from Michael Bickel today, who is working in the food safety space here in Zurich, and by Ella Sauerborn, who is a PhD student in Munich and is also a medical doctor working at the Technical University of Munich Rechts der Isar Hospital, where we have conducted some studies on human health. So briefly, what Michael's work was focusing on was saying, like we identify pathogen and resistances from whole genomes in comparison with established short-read sequencing approaches. These short-read sequencing approaches, they would normally be applied to the genomes that we have obtained from our cultures. But to obtain this data, often it can take really weeks or months because the sequencing is done in sequencing centers like outside of the actual like food production place or the clinical space, and until the data is generated and interpreted, yeah, very often it can take months. So he wanted to look into if a fast nanopore, like real-time nanopore sequencing, is now comparable with short-read sequencing. If these errors that I mentioned before have basically been able to be eradicated. And he has just finished a big study that I hope we will publish by beginning of next year, where he focused on more than 250, and he's just sequencing a few more, isolates from foodborne pathogens from all sorts of sources all over Switzerland. And he then looked into if the genetic diversity here represented by so-called core genome multilocus sequence typing that is used to understand pathogen strain diversity is identical between these two approaches. And he indeed found that due to the improvements of the nanopore technology and together with the improvements in AI, we now have like very identical gene diversity between these two sequencing approaches. He still found one exemption, which is a very specific epigenetic modification that was only found in 2 Salmonella isolates. And this really seems to be a very rare modification. And one example where we see, okay, like here we still have more sequencing errors using the nanopore sequencing technology because our AI-based base calling algorithms have not yet seen this modification and have learned it. So like I think with this sort of biological research, we can now really explore, okay, to what extent do we still have problems in the base callers, and we can use this newly generated data to further improve the base callers. I think it's actually quite exciting application of AI directly between the application and the model training. So in Ella's case, which I thought I would focus a bit more on today, she actually was able to directly in the clinic compare the established diagnostics that I showed you before, the MultiTOF and the BITEC2 approach, to the nanopore sequencing approach. And she applied this to more than 120 carbapenem-resistant Enterobacterales at the Technical University of Munich hospital. These were all of the carbapenem-resistant isolates from 2023, and we focused on this carbapenem resistancy because carbapenem resistance is on the rise. It's leading to increased mortality and more morbidity in patients. And it is also known to often be transmitted by plasmids. These are these mobile genetic elements that I mentioned before that can very quickly be transmitted also between bacterial species, and therefore really leading to really fast transmissions between bacterial species, but also between patients and patient environment. But I will talk about this briefly later. And yeah, basically, Ella found that just based on the genomic information we would obtain from nanopore sequencing, she could make all the correct antimicrobial resistance predictions in these isolates. In the case where the established diagnostics had actually found the responsible resistance gene, she was also able to find it. But in 24% of the cases, the established diagnostic approach had actually not found the mechanism of resistance. And in these cases, she was able to find alternative genomically encoded mechanisms, such as, for example, a different copy number of ESBL genes or a purine loss. In addition, with this information, we are really able to go in much more depth than established diagnostics. For example, we can really do microbial strain and plasmid typing, and then also decide where that resistance gene was located, like in the bacterial chromosome or on a plasmid. So let me show you two examples of that research briefly, which, however, have already been published / preprinted. So in this study, Ella looked at a patient who showed up at the Technical University of Munich hospital, immunocompromised with fever. And a Klebsiella pneumoniae infection was determined that was resistant to carbapenem antibiotics that we just talked about, but susceptible to the last resort antibiotic ceftazidime-avibactam. Now, ceftazidime-avibactam was administered to the patient, but the patient further deteriorated, and when another sample from the patient was taken many days later, the infection was still caused by Klebsiella pneumoniae, but the resistance profile had turned completely around and was now resistant to this last resort antibiotic. And while obviously the antibiotic administration was changed at this point away from ceftazidime-avibactam, the patient unfortunately passed away a few days later. So we were now able to apply nanopore sequencing to the very same isolates that had been subjected to the established diagnostics. And what we found is that for this second sample that was taken here, we found the very same resistance prediction because we found KPC14 genes that confer this resistance to ceftazidime-avibactam and no resistance to carbapenems. In the case of the first sample, we found this evidence of resistance to carbapenems encoded via the KPC2 gene, but we also found a single copy already of this KPC14 gene that actually confers resistance to ceftazidime-avibactam. Now, what is a quite beautiful application of nanopore sequencing in the clinic is that you can basically sequence in a fully flexible manner, meaning you could just have run the sequencing a little bit longer to obtain more data. And we simulated this like by just running the sequencing longer. And in just 8 hours, we found 4 more highly accurate copies of this KPC14 gene, meaning that already at this point where the patient showed up, we could have predicted this resistance to ceftazidime-avibactam. So what happened here? We can now, after sequencing, generate the de novo assemblies of both of the isolates, and we saw that in both cases we had the same Klebsiella pneumoniae strain, a high-risk strain, and that we saw exactly an abundance increase of the so-called INC-N plasmid that you see highlighted here in blue that encoded either the KPC2 or the KPC14 gene. But what we saw is that the INC-N plasmids that carried the KPC2 genes basically disappeared like in this time frame, whereas we saw like a significant increase of the INC-N plasmids carrying the KPC14 genes. In a way, a beautiful example of natural selection in that ceftazidime-avibactam was administered to this infection, and obviously then the Klebsiella cells with harboring like the resistance to ceftazidime-avibactam having an evolutionary advantage. So I hope that this shows you a little bit how at least the parallel application of nanopore sequencing in the clinic can really enrich information from established diagnostics. Now, obviously, this doesn't happen a lot, I have to emphasize, right? Like in the majority of the cases, established diagnostics already gives you the information that you want. But we do know that in many cases, we have to obtain a lot more detailed information. Or like the more detailed information can be helpful for actual treatment decisions. That's why I would like to show you one more example where this is not even about treating the infection of a patient, but like thinking a bit more about infection prevention and control in the clinic. So before infections might happen and outbreaks might start. So in this specific case, which is also pre-printed and which we actually just sent the revised version back to microbial genomics, so I hope this will be available in a peer-reviewed version very soon as well. We are looking at plasmid-mediated resistance spread in environmental reservoirs. So again, at the hospital in Munich and its hemato-oncological ward had a suspected carbapenem-resistant Citrobacter transmission between patients and environmental drains for more than 1 year. So these are the 14 samples you're looking at, 10 patient samples, 4 samples from the drain. And these 10 patients all resided in the respective rooms where these drains were found. And for over 1 year, there were like these Citrobacter infections popping up, so that the clinician suspected a link between those. But based on the established diagnostics that you can see here, they couldn't establish this link because the mass spectrometry results for the bacterial species identification only told them like a very high level classification, like that these species were part of a specific complex most of the time. The resistance, phenotypic resistance testing told them that there were like some shared resistance genes, but this is also a very high level classification of the resistance genes here. And obviously, the clinicians wouldn't know if these resistance genes were encoded by the bacteria, by plasmids, and if it was plasmids, if they encoded partially both of the resistance gene or just one, or, like all of this could have no link whatsoever, and there was nothing to worry about, right? So like basically established diagnostics didn't give the infection prevention and control unit a lot at all. With the nanopore sequencing information, we were able to generate the de novo assemblies of all of these different isolates. And we found that actually in terms of bacterial species, only 6 of these isolates were connected and were of the same strain, or so-called sequence type 91. And when we compared these 6 genomes that were connected with the global publicly available genomic data of this Citrobacter freundii sequence type 91, we found that they were actually really closely related, and they differed from each other by only a few core genome single nucleotide polymorphisms, or SNPs, meaning that they could all be like clustered together into the same like outbreak. I think particularly interesting here was that drain C and sample 1 only differed by a single SNP, which is really interesting because the patient 7 actually resided in this room C just the month before the sample was taken. So here we are probably talking about like an infection event. We don't know into which direction, but that there was a connection. And finally, and maybe I should say here, right, that the drain samples are really important because there is a fear of that these microbes can survive, especially in biofilm formations, in the sink drains and also other drains, and then lead to reinfection of patients, for example, via aerosolization or splashbacks. Yeah, and I think this is one event where it might be that we have observed like this, or obviously the infection might have happened the other way around as well, though, that the patient actually led to the infection of the sink in this case. Another example is that patient 2 and patient 9 and 10 clustered super closely together, only differing again by a single SNP, whereas they were actually in the same room, room D, more than 13 months apart. And having like this little genomic difference over this like large time span is very unusual, really suggesting that there was a biofilm of this pathogen somewhere in this room, not in the sink of the sink drain of room D, because we didn't find anything there, but somewhere else, that led to this reinfection 13 months later. But now let's jump on what we can do beyond the sequence typing, as we can also look at the resistance genes. And we found that all of the resistance genes were here encoded by plasmids, either INC-N plasmids that we have already heard about, or INC-L-M plasmids. And like just focusing now on this KPC2 resistance genes on the INC-N plasmids, we found that all of those that you see marked here actually were really closely related. So they differed by very little in terms of their sequence content and also structural difference, meaning you can again talk about a plasmid outbreak. Plasmids are inherently a little bit more difficult to deal with on a computational level because they can mutate really quickly, including a lot of structural rearrangements. But there are other tools, there have been tools published that we are using that are basically guiding a way forward, as here by Sam Iqbal's lab that was published last year. Like to tell you one interesting example here is, for example, the plasmid that we found in patient 1 that was very closely related with the plasmids from patient 8 and the zinc A. Here you can just see like the sequence similarity assessed by the mesh distance, and you see that the distance is like so small that this basically means these plasmids are very similar between 1, 8 and A. And this was again interesting because a patient A resided in room A, a patient 1 resided in room A 8 months before patient 8 resided in room A, where we are therefore quite certainly talking about a plasmid-mediated and environment-mediated transmission of resistance, which I think is a really important information for the infection prevention and control team, which since then have been able to, like, I mean, basically what they did is they didn't just clean the drains. They actually installed completely new bathrooms that would include like optimized drains that cannot harbor biofilms so easily. And I also always like to mention that in many countries by now on the ICU level, you don't have any drains in the ICU anymore in order to avoid such biofilm formation. So now going back though to what I talked about beforehand, that we can actually try to speed up our established diagnostic approaches significantly as well. Because like what I've shown you right now is the comparison of the established diagnostics and nanopore sequencing and application to the very same isolates or genomes. But obviously, ideally, we would like to skip this entire culturing approach, which is said is biased and lengthy and requires like a lot of specific like biosafety levels and so on. So we would like to apply genomics right away to the actual sample. And this is what's called metagenomics, that you just sequence like all the DNA in an entire sample without enriching for anything. Now, also for this application of metagenomics, nanopore sequencing can be very helpful. Firstly, because we know it is the only really rapid on-site sequencing approach that we have, but also because its potentially very long sequencing reads can be pieced, as I told you before, can be pieced together into the de novo assembly so that we can then do really in-depth pathogen and strain typing and also resistance detection. And that is really important because we know that there's often like a much more pathogen diversity in our original sample than in what's actually reflected by just one or a few isolates that we are sequencing back here. So that can be diversity between pathogenic species, diversity in terms of strain diversity, which might also be important if you actually want to trace an outbreaks, like I just showed you in the Citrobacter case, but also diversity in the resistance genes that they carry. Yeah, right. So before I actually dive into nanopore sequencing for metagenomics, though, I quickly want to show you just a few examples of my own research in the past and of the group now in terms of metagenomics, because here I think we really now embrace this One Health idea and have been applying metagenomics to a lot of different systems. So during my postdoc in New Zealand, where I worked with the National Department of Conservation, we applied nanopore sequencing to a lot of critically endangered species. And here just two articles in case you're interested, where we applied this to the kakapo, this critically endangered parrot species in New Zealand, like both for pathogen monitoring as well as actually genomic monitoring of the species itself. And where, because these guys live on really remote islands, we would actually really bring the sequencing technology in the field and showed that this could be of help for the conservationists. Yeah, there are a few other species that we looked at during that time. But we also, like I've also in the past applied nanopore sequencing to other, yeah, like let's call it One Health applications. We have, and that was actually as a side project during my PhD at the EMBL EBI, established nanopore sequencing for freshwater monitoring, which is a very old published study though, so, or it's from 2021, I guess it's old by now. And then in the group now, in my group in Munich, we have also had several applications. We have established nanopore sequencing for air monitoring in collaboration with the Global Health Institute in Barcelona, and also to monitor wetlands that we used as an example for ecosystems along the East Atlantic Flyway to see how migratory birds are actually transmitting pathogens and resistances. And here in Zurich now, we have started projects that focus on agricultural monitoring as well as on wastewater monitoring, just to give you an overview of what metagenomics can actually do to better understand One Health. I thought I really briefly talk about the air monitoring part, because I think that's something that interests a lot of researchers and also clinicians and other stakeholders for pathogen and resistance monitoring. So what we have done here, or this was research led by PhD student Tim in the group, he has optimized air sampling methods together with nanopore metagenomic sequencing to really have a rapid and on-site air surveillance approach. And here we are focusing on this liquid impingement-based approach using the so-called Coriolis samplers, because when we applied this one for at least 3 hours, we would actually get enough material from the air through the active sampling that we could like do this metagenomic sequencing, which, as said, is just based on sequencing on the DNA that you have in your sample. And we applied this approach to assess the urban air microbiome in the city of Barcelona together with our collaborators here. Here you see like the 5 different locations that we sampled, and also like sort of the anthropogenic impact here reflected by remote sensing applications. And we would sample air at these 5 different locations in a completely randomized manner, randomized in terms of daytime and also a day of the week across 2 weeks. And what we found is that what you can see here in this principal component analysis is principal coordinate analysis, sorry, is that we really saw the samples from these 5 different locations clustered together in terms of their microbial composition, meaning that this approach is able to really robustly assess a location-specific air microbiome. So if we briefly dig in a little bit, these here again are the 5 different locations and how they look like. We see that across these different replicates, we find like a really highly stable microbial composition. And this was really promising in the first place, because we obviously firstly want to see how robust our surveillance approaches are. We can then also look for the presence of antimicrobial resistances in this data. And for example, we found that in Poblenou in Barcelona, where we sampled actually close to a clinic, we found evidence of beta-lactamases that were chromosomally encoded by Stenotrophomonas maltophilia, which is one of these like really new emerging clinical pathogens. So we obviously don't know at all if this came from the hospital or where it came from, if it was infectious at this point and so on. But I think it really proved, okay, we can find relevant pathogens and relevant antimicrobial resistances. But now, coming back to this overview, right? I've shown you a few applications of the metagenomics, but in order to really be able to apply metagenomics, we have to again compare the approach to the established diagnostics, right? Like how sensitive we are, how precise we are, how specific we are. And this is obviously a little bit more difficult now because we are talking about a completely different sort of sample type. Yeah, here we are looking at the raw sample, let's say, and here we are looking just at bacterial isolates. But I do think that this comparison is important if we actually want to think about the efficiency of surveillance by metagenomics. So here we are coming back again to the food safety and human health space, where we at least have the option of doing this application of the established diagnostics. Now at Zurich, the food safety metagenomics work is led by Francis and Michael. Michael, you have already met before, who are currently developing protocols for focusing on monitoring Listeria foodborne pathogens, but that could obviously be applied to any other sort of foodborne pathogen, which I think is a very important pathogen because it's really leading to often deadly infections, and it has a very high strain diversity that often gets lost during the culturing process and can persist in a so-called viable but not culturable state, which is actually infectious but would be completely missed by the established diagnostic approach. I also like to mention that in food safety, also people started using so-called quasi-metagenomic approaches, where they are not isolating the bacteria, but they are looking, like during the enrichment process, how sensitive their approach is. But I don't think I will focus too much on this in this lecture in order to focus more on the AI in a moment. And then on the human side, we have PhD students Harika and Ella in the group who are focusing on the clinical side, again on carbapenem-resistant Enterobacterales, where we have now sequenced rectal swabs of patients as well as their genomes, and we have access to the established diagnostic results, meaning that we can really one-on-one compare how well metagenomics works for surveillance. And here I think it's particularly interesting that we can also look if we can assemble the plasmids that we know often carry the resistances in the carbapenem-resistant Enterobacterales and therefore predict the resistances of the pathogens. So now importantly, to think about really using metagenomics instead of established diagnostics, we have a few challenges left. And I would say these are the three main ones. And we have a problem with sensitivity because we're obviously going to sequence a lot of other things. We are going to sequence the host if it's like, for example, a sample from a patient, and we are also going to sequence general microbial diversity. In addition, just because we obtain the DNA or genomic information about a pathogen, it doesn't mean that this pathogen was actually alive at the point of sequencing, because we can't actually make a viability inference from DNA at this point, whereas if you can actually culture the pathogen, you know it was viable and growing. And finally, if we find resistance genes or plasmids, it is inherently difficult to associate those to the pathogenic host. Now, maybe I firstly want to say that with all of these problems, the long reads of nanopore sequencing that we already mentioned help. Yeah, because they allow you to better connect your pieces. However, I think in addition, nanopore sequencing provides like additional like advantages that, and this is where I started calling it like this is Squiggles coming to the rescue, right? Of like applying metagenomics for actual surveillance. So in terms of sensitivity, what we can do is we can use an adaptive sampling approach that is also very unique for nanopore sequencing, where we can basically tell our nanopore what DNA we are interested in. And if we start sequencing sequencing a DNA strand and we discover that we are not interested in it after a little bit of sequencing, we can just reverse our voltage and chuck it out. And I would really like to emphasize that this is like dependent on very fast, rapidly implemented AI models that can do the base calling and characterization really in real time, and then tell the nanopore that we would not like to sequence a certain read we are not interested in, which obviously effectively means you're enriching for what you are interested in. For the viability inference, so I think for the other two, I will briefly now dig into some more detail for the end of my, I think I have like 5 minutes left to really show you more direct applications of AI in this space. So for the viability inference, I had the thought a while ago that potentially we could infer signatures of degradation from our DNA using this raw nanopore squiggle data. And if such degradation could actually point towards if DNA came from an alive or a dead organism, because DNA from the dead organism would be exposed to external damage, it would lack any sort of DNA repair mechanisms. And so like the question was, or the hypothesis was, that we can identify these sorts of viability related signatures in the squiggle data. So before I come to the last point, let's briefly dig into this one. So this is also a study published this year by PhD student Harika in the group, where we exposed E. coli to UV damage, we obtained the squiggle data, and we would then train residual neural networks for a binary classification, dead versus alive. And what you can see here is that we firstly trained the model on chunk levels. So we had like the same sort of input length. And you can see that on the chunk level, we are obtaining really good area under the receiver operating curve, characteristic curve, and area under the precision recall curve. And if we then go from this chunk level to an entire read, so we are basically aggregating information by giving the entire read to the model, we can even further improve this accuracy. So have nearly perfect detection of viable versus dead. These are obviously the results of the completely independent test data, but still of UV-killed E. coli. Now we can apply explainable AI methods, such as class activation maps, to actually look what the model is finding. And here we see that our class activation maps find, and I hope you see this here, this like sudden drops in the squiggle data. And if we just define like a really simple rule to define viability based on these drops, by just saying we are having like a horizontal cutoff here of these sudden drops, we can actually also with quite good, not as good accuracy as the AI model, but with good accuracy predict the viability from the data. And these sudden drops probably have to do with the formation of pyrimidine dimers upon the UV exposure. So we can actually biologically explain why our model is able to do the right prediction here. Obviously, though, if we actually want to apply this to complex metagenomic data, we have to think about generalizability. So what we did is we applied this model across different taxa. So instead of applying it to E. coli, we applied it to Chlamydia suis, which is a very different pathogen, an obligate intracellular pathogen. We had two replicates here, both of dead and alive Chlamydia. And we see that our UV model works very well, meaning generalizability across different microbial species that have completely different DNA context is possible. Now, if we, however, look into a completely different killing method, and this here we used antibiotic exposure of two mock communities where we had susceptible E. coli that was susceptible to the antibiotic, as well as resistant Klebsiella oxytoca, we find that our accuracy drops. So in this specific case, all of the susceptible, or majority of the susceptible E. coli were actually still classified as alive, whereas they were killed by the antibiotic. So we saw that obviously we don't have this generalizability across killing methods. We, however, generated new clean training data and trained a model just on carpenicillin-exposed E. coli. So carpenicillin was the antibiotic we used here. We obtained a good model performance. And then also applied this data. So this is the performance on the test set, and applied this data to our mock communities and found that accuracy actually increased quite a bit. Not as good as the UV model, but it did. And if we look into the explainable AI methods here, we again see that for the application of the UV model to Chlamydia suis, it works well because it again identifies the sudden drops that are most likely being associated with this specific UV damage. Whereas for the antibiotic exposure model, we find a very different, a bit more complex, I would say, pattern of the class activation maps. And we haven't been able to dig a lot more into this, but I do very much believe that if we, as we know very well from natural language processing approaches of AI, we are able to diversify our training data more and are able to then use more generalizable models such as transformers instead of the residual neural networks that we've used here. I'm quite confident that we can bring this approach to the metagenomic level, but this is what we are currently doing now. So diversifying the training data to then be able to train these bigger models. I think this is just like the very first proof of concept paper that we published this year. So now, lastly, and this will be my last 2 slides now, Victoria, because I do think I should have gone a bit faster at the beginning to focus more on the AI part. So we like the last problem I told you about was about this resistance gene and plasmid associations. So we actually want to know, was a plasmid or resistance gene contained by a pathogen or not? So could this have health impacts or not? And here we started leveraging the methylation patterns that, as I told you before, we can obtain for free from the Nanopore Squiggle data using specific AI models. Now, this has not been our idea to use these methylation patterns. This has been encoded in a nice tool by other researchers, but I do think there are still quite some shortcomings of applying the tool as they've put it out. So we've started exploring this in some more depth. At any rate, like what is this based on? So like why can we use methylation patterns to associate, for example, plasmids with their hosts? This is based on the assumption that we have methyltransferases encoded in plasmids that would methylate very specific sequence motifs. And whenever like this sequence motif is not methylated, there is a fitting restriction enzyme that would cut at these motifs. Now, this is something that bacteria are using as an intrinsic defense mechanism by basically methylating their own DNA. But if then like foreign DNA, just such as from a phage, is coming in, they can efficiently cut it and protect themselves from this phage. But this also means that any sort of plasmids that swirl around in the same bacterial cell are also probably methylated in the very same way that the bacterial genome itself. So we started exploring this, and here Harika and Ella, which you've seen before, have started collaborating. And we first built like a mock community of different clinically relevant pathogens, where we then used the intrinsic methylation data using these AI-based base calling mechanisms. And would associate them with the specific bacterial chromosomes. And like this would be able to really associate the majority of the plasmids, which you see highlighted here in bold, with their respective bacterial chromosomes. Here the color coding is according to to where these different assemblies were coming from which species. So the only, and I take this short now, but the only plasmids that we were not able to associate correctly are these guys over here, and we are currently figuring out why this is happening. Like we are basically, what we are doing is based on these like methylation motifs, we are sorry, I'm being called all the time, but I should ignore this. Like we are clustering together the assemblies here just based on the root mean squared distance, and are able to associate the plasmids to their nearest neighbor. And we found that in this specific case, out of the 18 plasmids that we recovered, 14 were correctly associated with their host bacterial chromosomes. If we are only looking at quite long assemblies, categories. Nearly all of them were really accurately classified, which means the shorter ones might have a problem because there is just not so much methylation information. And finally, if we are in addition filtering for the presence of the respective methyltransferase, we can actually accurately predict all of the plasmids associations. Now, this is obviously a mock metagenomic data set, and we are currently applying this to the real data, the rectal swabs, where we know which pathogens are hosting which plasmids. But I think this is like some first really promising result. And with this, I'm happy to discuss especially the AI part, obviously, of the research in a lot more detail now, but would like to thank especially like my new collaborators here in Zurich, where I've shown the ongoing work, and then the entire group that still sits in Munich. And here are the most important publications. So thank you very much.